a few seconds and then we can go ahead. Christina, it might be already on your screen.
Yes, it is.
Perfect.
Okay.
Perfect.
So
you can go ahead.
okay, thank you. Well, I will start by thanking every attendee for being with us today. It's my pleasure to present mimsys G as a new, let's say, platform for 3D cell culture. I will start by making a quick preview of what the webinar will focus. So we will overview a little bit 2D versus 3D cell culture. We will check some of mimsys G features. Then we will look a little bit how to encapsulate cells within mimsys G. And then I will present some data, some of the performance of cells in 3D with mimsys G, cell viability, histology, and then end with some further applications.
Christina, 1 second. Forgot to mention that if you have any questions, please use the chat system. Send us the question and we will answer everything at the end of the webinar.
Okay, so I can continue.
Yes.
Okay. Well, I could start by showing a statement from a report published late last year by Data Monitor Healthcare, a report called 3D Cell Culture, where they stated precisely that 3D culture has the ability to translate from bench to bedside in ways that 2D never will. And at Irisbiosciences, we truly believe in this. So we are making lots of efforts to provide tools to better help scientists to perform 3D cell culture. We all know that currently 2D cell culture is the standard. Cells are grown in 2D surfaces, but this doesn't really mimic what happens in human body. Cell response might not be as natural as in the human body, and the experimental results might be quite unrealistic. We believe that if we provide a 3D environment for cells where they can sense the environment in every way all around, this could mimic the body-like environment, and therefore the cell response could be closer to what it would be in the natural context. And then at the far end, the experimental results will far be more realistic. So what is mimsys G? It is definitely defined. So its composition is solely methacrylated gel and gum. So gel and gum, as the backbone of mimsys G, is from a natural source. It's a polysaccharide, and it's already FDA approved as a food gelling agent. Most of its benefits is that it does not contain any animal components. There are no growth factors or proteins within mimsys G, and there is no toxic compounds. So because it is defined, for sure we can warranty batch-to-batch reproducibility. This polymer is water-soluble, and you can perform the entire protocol of mimsys G preparation and cell encapsulation either at room temperature or at 37 degrees to mimic the physiological temperature. And gelation is really quick. In 5 minutes you can have 3D hydrogels. Well, after gelation. So because mimsys is so defined, the user can control every single supplement of the cell culture. So there's no unknown proteins or growth factors that can interfere. The production of the hydrogel, so every sample that you are producing, is very consistent. The hydrogel is pipettable, and at the end you can have very reproducible experiments. So every experiment that you produce is very reproducible. And the whole setting to produce the hydrogel is very polypeptide. I will now show a video where we demonstrate how to encapsulate the cells, so you can more easily see how the procedure happens. So each vial is provided sealed and sterile. It has 200 milligrams of the polymer, so you just need to add 9 milliliters of deionized water. You should then close the vial and mix. This can be performed at room temperature, or if you prefer, it will be more quickly if you put in a water bath at 37 degrees. In the meanwhile, you can prepare your cells. So if you have adhered cells, you can take your time to trypsinize the cells, to count the cells. I definitely recommend to check viability before you encapsulate. You can use trypan blue. And then you should calculate what density of cells you want to use. So you can encapsulate from 1 million cells per milliliter up to 10 million cells per milliliter. So once you decide, you put the corresponding amount of cells in 1 milliliter of cell culture media. So this will be enough to actually cross-link the gel. So when you pipette the cells in the suspension, the 1 milliliter suspension, just add to a mimsys G vial. I recommend to mix by swirling, not so much by up and down, to avoid losses of the hydrogel. Then, if you want to work in a 96 well plate, you just need to pipette the solution in the desired volume. You can work with 25 microliters or up to 150 as far as we have tested. And you can for sure use other well plates that you desire. At the end, you just need to add culture media on the surface of the hydrogel, and this would be enough to cross-link the hydrogels and the cells within it. So when you're done, you just need to take your plate with the culture media to the incubator, and let's say in 5 minutes the gels are fully reticulated. You can leave your plates within the incubator, but if you wish, you can take them out and perform any kind of assay. We have prepared some protocols to help scientists to work with mimsys G. So the very first protocol of the handbook is a protocol to explain how to encapsulate cells in the hydrogels. So it's very simple. The entire procedure is in the protocol, and we can provide this. So what are some of the mimsys G key features? One of the most important is its mechanical properties are tunable. So if you work with cells that require a more stiff material environment or a softer environment, for instance neurons, you can tune these mechanical properties by working with the density of the material or with the cross-linker. You can use divalent cations to increase the cross-linking, or you can even add photoinitiators to the solution and cross-link it by exposure to UV light. And here is a plot just to show some examples of differences of mechanical properties with different cross-linkers. Another feature of mimsys G is the fact that it is injectable. So it has such a viscosity that allows delivery of the material by injection, and you can inject either in studies in vitro or, most importantly, in vivo situations. And therefore you can control either the spatial and the temporal gelation of mimsys G in situ. And this is a quite interesting feature. Another interesting aspect of mimsys G is that the hydrogel is fully transparent. Here we show an example of a 7 millimeter diameter and 2 millimeter height hydrogel. So as you can see, it's really 3D in terms of height. And when we have the cells encapsulated, as we can see here through this image, we can, simply by using bright field microscopy, observe the cells within the hydrogel. And the hydrogel is in the well plate with the culture media, so there is no need to transport it to another system. We have tested several cell types within mimsys. We have encapsulated cell lines, namely L929. We have tested some primary cells, such as chondrocytes. We have encapsulated adipose stem cells. And we have tested also non-adherent cells, such as the stromal vascular fraction. And here I take the chance to show that we also can perform fluorescence stainings. So as we can see, we have examples of cells that were stained within the hydrogel with DAPI and phalloidine to show cell nuclei and the cytoskeleton. Or if you even want to do live dead, you can perform calcine and propylium iodide staining to see live and dead cells within the hydrogel. We have tested also some different cell densities within the gel. So we've tested 1 million cells per milliliter, 5 and 10, and you can see the differences directly in the hydrogel. And we also prepared protocols to help the user to perform these stainings with the hydrogel. So one of its key features is actually that the hydrogels are compatible with microscopy, either bright field or fluorescence stainings. Here we demonstrate an example of cell viability within mimsys. So we performed live dead fluorescence staining. So calcine is shown in green, showing live cells, and dead cells, the nuclei would be stained by propylium iodide. And here we have an example of adipose stem cells encapsulated at day 1 and also after 21 days of culture. So we have very good viability within the gel. Here we also have a protocol to help preparing the live dead assay. And also to check cell viability, we commonly perform MTS assay. So MTS is a cytotoxicity test used in the ISO guideline 10993. So we use this to check cell metabolic activity within the hydrogel. On your left, you can see that we encapsulated different cell densities within a 50 microliter hydrogel, and we can see that metabolic activity is proportional as we increase cell density. And on your right, we can see the progressive viability of cells in a long-term culture. And in this case, we're showing metabolic activity of adipose stem cells at 10 million cells per milliliter up to day 21. Also, we've tested diffusion of MTS in different sizes of the hydrogel. So here we have an example of hydrogels of 25 microliters, 50 or 100. And when we have 5 million cells per milliliter of density, we see a progressive doubling of metabolic activity as we double the volume. So we have no problems with diffusion of the metabolite throughout the hydrogel. Here we also have an example of when we have the same amount of cells in the hydrogel but in different volumes, so in different densities, we also see that the absorbance that we read is very similar despite the volume in which the cells are in. So we can state that, once again, that the volume of the hydrogel doesn't influence the metabolite diffusion. For this, we also prepared a protocol, and with this we might state that mimsys is compatible not only with microscopy to have qualitation data, but also with quantitation. So you can actually quantify some of your outputs of the cells with mimsys. And mimsys is also compatible with histology. So we have performed several histological analysis by fixing mimsys hydrogels in formalin and then simply embedding in standard paraffin embedding systems. Then after sectioning, we can stain as any other tissue for H&E, for instance, or any desired staining. Here we have two examples of an in vitro staining of the cells within the hydrogel, or an example of an in vivo sample, where we have mimsys quite nicely integrated with the native tissue. As the last protocol, we prepared a very simple protocol for histological analysis, and stress out that mimsys is compatible with standard histology. To end, I would leave some of references. So where mimsys has been used already in the literature, we have some publications where we demonstrate in vitro and in vivo biocompatibility of mimsys, some of its mechanical properties, injectability, and long-term cell culture. And also a very nice study where mimsys was applied in vivo for disc regeneration purposes. Although we haven't yet explored extensively, we believe that the properties of mimsys are compatible with automation and, for instance, 3D printing if required. So this is something that if scientists are interested, we can further speak with each other and try to find a way to explore these applications. So this is a slide just to summarize the major characteristics of mimsys and its applications for 3D cell culture. So first of all, it is xeno-free. It's nutrient permeable. It's transparent and injectable. And gelation occurs very fastly in 5 minutes by ionic cross-linking. Some of its benefits is that the polymer is highly reproducible, no batch-to-batch variability. It's very easy to handle, and you can customize the hydrogel stiffness according to your cell's needs. And of course, it's compatible with several cell types, such as stem cells and cell lines. I recommend that you use our recommended protocols. I think we prepare them to help the scientists to easily work with this new product. We have tested long-term culture up to 21 days, but we can help or advise if you desire to make even longer cell culture periods. And we have also already some expertise with in vivo experimentation, and we will also be happy to help in any doubts that you might have or compatibilities of the system with your experiment. So this is all for today. I thank you once again for taking your time to learn a little bit about mimsys. If you have any questions, please feel free to contact us through our tech support, and feel free to browse throughout our website, which is Irisbiosciences.com, where you will find most of the information that I presented today, as well as some other products that we are providing. So thank you very much.
Thank you, Christina, for this wonderful presentation. I will send the record of this presentation together with your wording as an MP4 file to every attendee, so you will receive it tomorrow or the day after, and you can look it up, and then you have also the email address where you can send your questions in to, and so on. So thanks again for attending our webinar, and I hope you enjoy